Okay, thank you very much for the invitation to present my work. So I work with the Structural Genomics Consortium. I've been at several sites, but now I'm again based at the Karolinska Institute in Stockholm. And I'm going to present our efforts in antibody validation and more specifically our IPMS method. So first, I just wanted to take the chance to introduce the SGC to those of you who don't know who we are and what we do. So we are a public-private partnership and started in 2004. And we have funding from several public sources in Canada and in the UK, and also now the European Union that allowed us to reopen a site in Stockholm where I work. And we also have 8 major pharma companies that you can see down here. And we have many sites today. The major ones are still Oxford and Toronto, but now also a small site in Stockholm, Campinas in Brazil, Zurich, North Carolina, and Germany or Frankfurt. And what do we do? We are an open source research organization, which means that we are promptly placing all the results that we get in the public domain. And very importantly, we have promised to never file patents, which makes it much easier for us to work with a lot of pharma companies and academic collaborators. So the focus areas then, of course, you can guess from the name, Structural Genomics Consortium, we solve structures. So that's the main pillar, and it's been since the start, and we have now more than 2,000 structures in PDB. Until 2015, this was a number, and we are still continuing to do this, obviously. Chemical probes started in 2007, so we are using our proteins in screening campaigns to find small molecules that can then be used for target validation. We call these chemical probes. Renewable antibodies or biological probes, a program that was officially started in 2011, although we did pilot projects from 2008. And I have been managing these from the start. So this is my area of expertise, which I will go into more detail. And a novelty from last year is that we now also do cell-based assays on patient-derived cells. So I have colleagues in my lab at the Karolinska who specifically do this, where they test chemical probes, and we will also test antibodies. So these are the labs involved in the EU project that I work for. So what does the SGC actually do? What is our mission? Well, if I should summarize it, it's really to boldly go where no one has gone before. And I'm going to illustrate it with this picture. So this is called the Knapp graph, named after Stefan Knapp, who is the PI for kinase work within the SGC. So what he discovered was that of the 500 kinases that we have in the human kinome, most people are working on the first 50. Here is citations in PubMed, so here's where people publish. But studies have shown that actually all of them, or the ones that would be interesting in drug discovery, actually evenly spread out all over the 500 ones. And if we look at what companies do, where they file their patents, it kind of follows the same pattern. It's the top 50 that gets most of attention. And you could summarize this in that actually 90% of global research focuses on 10% of human biology. And you would think that it would change with time, because these are old numbers. But then again, a few years ago, we redid this, and we saw again that the pattern stays the same. Here is where people do research. And this then could be called the unknown kinome. So this is what we are targeting at the SGC. We're a large research organization. We've got a lot of financial muscle compared to academic groups. And we don't have to develop a product directly like a company and get revenues back. So we can afford to go in here. And this is what we're doing. So we try to work on things that are kind of unknown and bring out research tools that people can use, like chemical probes and antibodies. And this is what we see, that whenever a new tool becomes available, then people start publishing. So antibodies then. Why do we work on antibodies? You would think that's pretty far away from solving structures. Well, what we realized was that we sit on a gold mine of purified proteins, and those could be used for more than just solving structures. For example, as antigens to select recombinant antibodies. So what we do then is that for select targets, we produce recombinant antibodies and we put them out there without restriction on use. And what we also wanted to do, this are kind of thoughts from the pilot project that was started in 2008. At the time, there were definitely not very good antibody validation methods in place. We have seen at this meeting that there are a lot more now available. But at the time, it was pretty poor. So we wanted to work on something that could be put to general use by a lot of people. So just to mention what the criteria that we are working against is. So for us, the antibodies has to have an affinity of at least 20 nanomolar. We want them to be able to IP endogenous full-length targets from a cell lysate, for example, HEC293, or other relevant cell line. We want them to be specific in our IPMS assay, or at least we want to know about the cross-reactivity. And for the biological probes that we are also making, they have to be having a measurable activity on cells in a suitable assay. And this is for these patient-derived cell assays that I was speaking about earlier. So they have to be against extracellular targets. And then, of course, neutralizing antibodies. We need to show that they can block the signaling activity. So, SGC proteins, as I mentioned, we sit on a gold mine. We have several thousands of purified proteins. And the good thing is that the protein quality needed for phage display selections is pretty much the same that is needed for crystallography. The only thing is that we need to be able to immobilize our proteins. So we put an AV tag on them, that's the only change. And then we in vivo biotinylate that during production. And then we send away the proteins or give them out to a phage display group. We have been collaborating with several different groups over the years. Sertev Sidhu, Koziakov, and Koidi for Fabs in an earlier project, and also Stefan Dubel at the time. Now we are mainly collaborating with Helena Persson in Stockholm at the SciLife lab. And Helena Persson has made this synthetic single-chain library that we're now using. It's called the SciLife lib, and it's kind of open access, which is very nice because normally phage display libraries are heavily protected by their creators. But Helena Persson is an incredibly open person. I'm very, very happy for this collaboration. So I'm not going to go into any details. I just wanted to mention that it's a synthetic library, and it's the diversity in the CDR loops is mainly based on tyrosine and serine and glycine, which was research made by Dev Sidhu at Genentech. So the first antibody project that I managed, I won't go into a lot of detail here. I just want to mention it so that you get an overview of how we work at the SGC. So we produced the antigens at SGC Toronto, where I was at the time. We then sent out antigens to the Fab labs. They did the selections and performed primary validation and then sent clones back. We recloned them and produced them as biotinylated Fabs in E. coli. We gave them to a validation lab also at the University of Toronto, led by Jack Greenblatt, where they did cell-based validations, mainly IP of various sorts. We then reformatted to IgG format for the best ones, produced them in hex cells. We revalidated the IgGs, and this project was supported by Life Technologies, which was the name of the company at the time, now Thermo Fisher, as I'm nodding at Matt Baker over here. So the validated IgGs and Fabs we then sent to them for putting them in their catalog so that they could be distributed. And if you want to know what targets we worked with, they are available at this link. So the novel workflow that I have now is simpler because we do more things in-house. So you can see all the way from antigen production to IPMS validation and a lot of biocore. We have not started to convert anything to IgGs yet because we don't think it's needed. We actually think that single chains and Fabs are just as good for most applications that you want to do for research. But we will probably go here as well for select antibodies. So the biological experiments that we want to do, for the biological probes, we want to put them in our cell-based assays on patient-derived cells. We're also doing co-crystallization, where we want to solve structures. And then, of course, we do have collaborators that we will give antibodies to. And we currently have no distribution channel. We are discussing with several. maybe Thermo Fisher, our old partner, maybe others. So this is still not really known, but it's important, I think, because many good antibodies made in academia are not so easily accessible for people doing research. So you can sit in a gold mine of antibodies, but if they don't get out there to research or use them, they won't be doing humanity any good. So this is important for us. So validation then, this is the topic of my talk, so what I should really be focusing on. As I said, at the time in 2008, the problem that we saw was that there was really no validation standard out there, and a lot of antibodies did not work as promised. So we wanted to establish a general package that more or less anybody could use. So the primary validation, as I mentioned, we used a lot of ELISA and also HTRF. We have now started to use BioCore in high throughput format, which means we just test every candidate from the selections directly on the supernatants with just one concentration to see if it seems to be binding or not, because it gives us additional information so that we can cut down the number of hits that we get with these methods. And then we do IP. It was chosen as a standard method. We have been running it in two ways, IP Western, which requires overexpressing cell lines. I will explain this on the next slide. And then IPMS, which has become our favorite method. And the good thing is that it could be done with any cell lysate on endogenous target protein. Follow-on methods have been chromatin IP, especially for proteins that we've been working with involved in epigenetic signaling, because they are obviously linked to chromatin, and also immunofluorescence. And because it's been a lot of talk about Western blot at this meeting, we don't do Western blots simply because our antibodies just don't recognize any denatured protein because they are all made against well-folded protein. So that's a limitation. If you want to do Western blots, forget about our antibodies, but else they work really well. So IP Western versus IPMS. IP Western, as I said, we do a pull down from cell lines overexpressing a tagged full-length protein. And this is important because what we do then is a secondary step where we do a Western blot detection with anti-flag. So this is the tag that has been used in the Greenblatt lab, and you can see it has a triple flag here. IPMS, we do a pull down from either from the same cell lines or from just a regular lysate of a cell line. We do trypsin digestion, and then we detect with mass spectrometry what came down. So basically what it looks like is this. We incubate antibodies with a lysate. We immobilize the antibodies on a surface, usually some beads. And it will then, of course, bind the bait. And it could also be one or more potential preys. And I should mention that the drawback is that it's very difficult to distinguish between these two, which I will come to in a little while. And then for IP Western, as I said, we need to work with a bait that's flag tagged so that we can detect it with the anti-flag antibody. For IPMS, we elute what has been pulled down, we digest with trypsin, and we detect and identify the peptides with mass spectrometry. So the mass spectrometry analysis, I'm not a mass spectroscopist, so I won't go into any details about how we look at the peptides, but basically, an important thing when you set up your mass spec method is that you need to know what type of samples you're having, if it's a single protein that gets degraded by trypsin, or if it's a lysate with a lot of different proteins that gets degraded, or in this case, we have an IPMS sample, so you have pulled out something with your antibody, so it's just a small part of the whole lysate, and then we digest that. So we're running liquid chromatography ESI MS, usually an Orbitrap. So the first thing you do is on the LC step, you separate what you're having in your sample, and then you will run it through the mass spectrometry, and you will get a spectrum that looks like this. And then you go in and analyze the different peptides, you map them onto proteins, and you will then get an output in spectral counts. So we compared a lot of IP Western and IPMS results for more than, or I think almost 1,000 different proteins. So I'm just showing an example of a couple of antibodies. So we had 2 antibodies against CBX3. You can see one of them worked nicely, the other one did not in IP Western. When we ran them in IPMS, the first one worked beautifully, both in just regular HEK cells. It gave a pretty low signal, but still it was significant, and then a very strong signal in the overexpressing cell line. The other one gave a low signal in both, but it kind of correlates with not seeing a band here. For CBX5, we had one antibody here. It worked beautifully in IPWestern and in IPMS. And as I said, we compared a lot of samples, and we concluded that they correlate very well. But IPMS is definitely a much more high-throughput validation method. So, going into data analysis. This is, whenever I get results, I get so excited. It happens a few times a year when we have had a screening round that I get to dig into this, so I really like it. The problem is you get so much data. So here is results for 30 different antibodies that we analyzed. And as you can see, for each run, we could detect in average about 800 different proteins. But the good thing is that all samples are of the same type, so they are controls to each other. And we could see a common background of 228 proteins out of these 800 that we know then are probably not specific because they appear in every single experiment. So basically what you look at when you look at IPMS data is that there is a lot of background noise, all this gray stuff here, and then you look for something that pops up specifically for one of your antibodies or for several if they are against the same target. So the first thing you want to do is to zoom in on your bait. So like, for example, in this case, we have 3 antibodies against WDR5. So we're looking then at, do they bind WDR5? So if we just concentrate on this graph, this shows then 30, the same actually 30 different antibodies as I had on the earlier slide. And then the result for WDR5, and you can see that it's a low baseline here. And then for the 3 antibodies that are supposed to bind this target, we see a good signal, and then it goes back to baseline again. So the good thing is that all samples are kind of controls against each other. But of course, if proteins are very similar to each other, like you can see here at the bottom of the list, we have tankerase 1, 4 antibodies, tankerase 2, 3 antibodies. So these are the 4 dots for tankerase 1. Well, 3 of them binds the targets, the 4th does not. But then for tankerase 2, we also see a signal. So it's an obvious cross-reactivity because these proteins are very similar. Another interesting thing is that we can also look at known complex partners to see if they come along. And here is again WDR5. It's known to be in a complex with RBBP5 and NASH2L, so we looked at those 2, and you can see that they both come up, and they are both specific across the panel of 30. So no signal for the other antibodies that were raised against other targets, but good signal here, and they kind of follow each other, so the one top here is top in all. So what if the bait cannot be detected? Well, obviously the protein might not be expressed. So you have to check this first. There are a lot of databases and publications available where you could look at. You could basically do this with any cell type. So just find something that's expressing your target, or you could try to overexpress it, like if you make a cell line for that, or you could even spike in your targets. I do not like that method, but I know a lot of people do spiked IPs. Another thing that might explain why you don't see a signal is that the epitope is not available. Most of our antigens are produced in E. coli, and of course that protein might look dramatically different than the full-length target from an endogenous HEK293 cell. So there might be post-translational modifications or a protein complex masking the epitope. So we have a success rate of about 2/3 of the targets will get an antibody that works for IPMS on HEK cells in our pipeline, even when we had working antibodies from the primary validation. Or the antibody is simply not working. Okay, so forget about that one. If you do a lot of antibodies, you will always find ones that don't work. So that was the bait. Now we try to look at the full picture. So I call this the hit list. So this is an antibody that was made against BRD4, and we are lucky to see that BRD4 comes up on the top of the list with 100 spectral counts. But there are also other proteins that show up here. So then we need to look at, are they kind of common contaminators, frequent flyers, background proteins, so to say, or are they actually something potential complex partner, or are they something that the antibody cross-reacts with? So for example, this one, RNA binding protein 10, it shows up in all samples that I run. So I know it's not specific. But looking down here on the list, BRD2, aha, very similar bromodomain, comes from exactly the same bromodomain branch on the tree. So this is probably a clear-cut case of cross-reactivity. And this is easy to spot because it's a very similar protein. So the difficult thing is to spot cross-reactivity when it's not something you expect. So you need to compare a lot of parallel samples. So here is a type of list that I normally work with. I get a ton of Excel sheets that I'm sorting back and forth again. So I reduced this one so that it should be easy to see on the screen. So in the beginning, I had 32 samples, I think, in this list, and now I reduced it to just 8. But here I have been looking at the antibodies against SMARCA4, and I sorted on this column, which has clearly the best binder with 60 spectral counts against SMARCA4. So the difficult thing here is what shall we do with these guys? How shall we look at this? So you need to reduce the list and remove everything that's not significant. So what we're doing is that we try to find the rows that seems to be specific. So this one, for example, it has 23 here and then a 0 all over. And you have to believe me now when I say that for the other samples in the panel of 32, the 0 was consistent. And here is another one of those lines, another one, etc. So what I did was I reduced the list and I removed all the rows that seemed to be unspecific. And what I came up with is this. Okay, so now we need to look at this. Is this biologically relevant? Is it cross-reactivity, or what could it be? So looking here, you can see that many of these belong to the SWI/SNF complex, SWI/SNF complex, SWI/SNF complex. Actually, the SWI/SNF complex looks like this. And you can see that we pull down almost every single partner of this complex, so SMARCA4, SMARCC2, ARID1A, etc. All of them are here. And here we knew the answer, so it was easy to see. And actually, it's also very consistent that the antibodies that work this well, they always have very good affinity. This one had a KD of 4 nanomolar, and these other 3 were much worse. Actually, I shouldn't have put them into IPMs to begin with, because I know they don't work if they don't have a pretty good affinity. But we did it anyway because those were the best binders we had for this target. So removing the background, another way you could look at it also is that if you know what your complexes should be, you can put a cutoff where you know you will retain your complexes and remove all the background. So this is something we did on all the fabs from Toronto in a paper that we published. So this is just a list of all the frequent flyers that we had, or not all, but the worst frequent flyers, I should say. You can see that they occur here, this one in 83% of all IP mass spec samples against fabs. And the percentages are high here. So we put the cutoff at 8%, and for that project, it worked really well, and it was based on data from known complexes. You could also visualize your interactions, not only through Excel sheets, but also through pictures like this. So here we had antibodies against CBX5 and CBX3, the ones that I showed the IP Western data for before. And what we saw for these was that a lot of known complexes, everything that's connected with red lines here, we could detect. We also saw some new partners that we don't know if they are real or not, and then we need to go in and look at that. So you really create a gold mine of data that you could look at. So to summarize this method, we have a very low experimental variability because we're using a lot of antibodies made with the same scaffolds. And all the antibodies have been pre-validated. And as I said, we are a little bit liberal with this. I would want the affinity to be at least 20 nanomolar because I know they are very unlikely to work if they are not so good binders. But sometimes we try it anyway. And we have standard protocols. And what we then have proposed in this paper that was published last year was to define IP gold standard antibodies, those that will precipitate the native antigen from the whole cell lysate, and where the antigen or a member of its known protein complex is the most abundant in the immunoprecipitate. So just like the SMARCA4 that you saw on the earlier page, it was on the top, and then we could see the complex partners. And we, in this paper, we presented results that were from samples that were sent out to 5 labs. And, well, I'm not expecting you to look in the details here, but the interesting thing is that we sent out 20 fabs and some lysate and an SOP, and then we asked them to do this and process their own data, and the results were very similar. Not identical, but similar. But you always get a lot of background from your instrument and your specific setup. So I realize I need to move on. I just want to mention that we have been doing a bit of immunofluorescence as well, but it's a very tedious method, labor intensive. But what we have been doing then is that we've been comparing our fabs. When we did have overexpressing cell lines, for example, we could compare with anti-flag, or if there was a commercial antibody, we could compare with that. Of course, you want really to have a good knockdown control, so we also did a lot of that. So we tested our fab with a negative short hairpin RNA, and then we did it with a specific one, and we saw that the signal was knocked out. And our statistics, I was asked this, so I just want to mention this, that out of all the antibodies that we tried that had all been pre-validated in IPMS, 76 of them, 76% of the gold standard fabs also worked for us well in IF. Actually, some of the inconclusive ones too, which means that they bind your target, but maybe something else too. We don't really know that yet. So that's why we didn't declare them gold standard. We've also been doing a bit of CHIP. Both pre-study was usually CHIP Western. We do it like the IP Western, but we do it with a cross-linked antigen, just to see that our antibody could recognize the cross-linked antigen. So we get a very similar picture as the IP Western. Then for selected targets where we knew the loci, we could then use CHIP qPCR to investigate enrichment. And here's an example of PRMT5 where we got a very good signal. And the statistics were pretty good, although we only worked for targets where we knew we had good antibodies and where we knew the loci. So 9 out of 10 of our antibodies worked there. So just to summarize then, I hope I've been able to convince you that IPMS method is very useful and that it gives a lot of information. I really like that you have the target IDs and not just a band or a dot or a spot in some assay. But you have to have multiple samples, because otherwise you won't get your background, which is so important to be able to analyze which signals are real and which are not. The good thing is that the method can be used to detect proteins from any cell type, and more medically relevant ones would be PBMCs, for example, which we are now moving into. I really hope that there will be a common database with results from various cell types shared. We are discussing this with Antibodypedia, and I really hope Thermo Fisher will help drive this development, because I know you're into a lot of IPMS. So Matt is nodding. I take that as a good sign. And I really want to, again, state that I think that IPMS is a really good primary validation method. It's high throughput, you see a lot of things, and then you can move into more labor-intensive methods like CHIP and IF and others. And with that, I would like to finish and acknowledge a lot of people at the SGC. I won't mention any specific names, but we are a large consortium. I've had lots of good collaborators over the years. Maybe SciLife Lab and Helena Persson and her boss Per Arvidsson, who are absolutely crucial for this. So I'm happy to take any questions. Thank you.
Thank you very much, Susan, for a great talk. Any questions from the floor? So, Mike.
I thought that was very interesting. I remember your first paper. I don't know whether those recent results you've shown have been published yet, the ones with the antibodies you've seen. But what struck me was this high similarity between what you're finding and what protein arrays can find.
Mm-hmm.
For instance, we also have a high level of proteins which we see every time, rather like your background, although reading the list, there were different ones. Mm-hmm. So we have similar sort of issues. It might be interesting actually to compare some of your antibodies, what they bring down by AP with what they see on arrays. I was thinking about what's the sensitivity that you're looking at, because mass spec is often regarded as a less sensitive technique than immunoassays, And I wonder if there are proteins which you're not seeing simply because of the sensitivity of the method. Do you have any thoughts on that?
Yeah, I can't go into any quantitative numbers, but we definitely have issues that we are looking at some proteins that are expressed at a very low level in HEK cells. So then we need to obviously find a way around this. Either, as I mentioned, a cell line that has a higher expression level, or we need to maybe produce a cell line that is expressing this target at a higher level, or just try to increase the signal by putting more cells in, and you know. So this is something that we optimize for our regular proteins. We haven't seen any problems, but we are currently looking at a few that we have problems with. So. That needs to be sorted out, but I would say we are on a good way there. But I would be happy to collaborate with you on you know, doing protein arrays. Yeah, exactly.
Your new library, are you using that as a way of producing chaperones for crystallography at all? So you say a lot of the proteins come from crystallography. Do you then get requests to try and make antibodies to maybe change a space group or change the structure slightly so people can soak compounds, for example? Have you used that new library? And do you find that there's any technique that's predictive of crystallization at all?
We're actually having a project now together with the crystallographers at SGC Oxford, where they are looking at our single chain framework, and they try to introduce mutations to see if they can improve crystallizability, because that's, I would say, the major drawback with co-crystallography with antibodies is that they don't always crystallize so well. So you have to try a large panel of antibodies, to my experience, to find one that crystallizes well with your protein. But yes, we do have a lot of requests from the SGC internally. Another thing also is that we're trying to increase complex size so that we can do cryo-EM, for example, on proteins. So if you add a Fab, you get an extra 50 kilodalton, and then you get over the threshold, which I think is currently at 180 kilodaltons or something.
How much do you find you have to screen, and what's the kind of hit rate target to target? Is it just a question of screening enough clones, or at some targets, like what percentage do you normally find kind of amenable to that chaperone type technique?
I cannot really say, because I don't have enough statistic data. We like statistic data within the SGC before we go out with statements. But it's pretty difficult to co-crystallize with antibodies. It's not something that always works. And the experience that I have and that other people have gained over the years is that you usually need to have a panel of different binders, and hopefully also of different types. So maybe single chains, Fabs, DARPins, or whatever you can get. Nanobodies, for example, have been shown to be very successful for membrane proteins especially. So I'm sure we are learning more. We've actually been discussing for years that SGC should do a large statistical study on a lot of targets with binders of various types to once and for all determine if one scaffold is better than the other for this purpose. We still haven't come around to do it, but I take this as some incentive to get going.
Thank you.
Maybe I missed that part, but could you remind me how you distinguish signals coming from components that precipitate as a complex with your protein of interest from non-specific background?
Yeah, so that is kind of the key thing here, that first, we don't know where the proteins in the immunoprecipitate come from. We don't know if it's the bait for the antibody that bound them, or if it's praise, so the proteins that came along bound to our antigen, or if they bound to the antibody itself. So that's the thing. So when we look at this enormous list of data, what we need to do is we try to find what seems to be specific for these antibodies. And if it's something that bound to the antibody scaffold itself, it will show up in all IPMS samples. So then what we do is that everything that comes out in every sample, we just see this as insignificant, and we look for the ones that come specifically with this antibody. And if we see the bait, like with this marker 4 with a very strong signal, and then a lot of other components specifically for that antibody, that's a very good indication that these proteins bound to our bait.
And one little question related to that. How many samples do you think that the lab needs to run to have a reliable
I knew I would get this question. Well, actually, we started to run 32 and 32 in parallel because that's a number that's very easy for my technician to process. And I would say that in our set of 32, yes, we see what's significant and what is not. If you go down, maybe you could get away with, 10 or 20, and you would probably see what's pretty significant. But of course, the more samples we do, then we can also compare sets to each other, even if the background could change slightly from set to set because the instrument might have been running other samples before that contaminates your current run, etc. But still, the more data you generate, the better your background will become. Just like any data set, the larger it becomes, the more reliable the statistics. Okay, thank you.
So now it just comes to the closing remarks.
To wrap up, yeah.
The wrap up. So Andy and I were just reflecting in the break that actually this has been from our point of view, a really great meeting. I think there's some common themes that have come out. And I think Jim, in his talk, just in the previous session, really summed up one of the key messages. And that is to think about not how your antibody has been validated, but what has your antibody been validated for. And the other thing I think that came out in actually our very first talk, Alison's comment about training and about sorry, can you all hear? I'm not quite loud enough probably. Alison's thought about training, and it's great to see the EuroMabNet really investing in their young researchers about awareness of what an antibody should be used for and how it should be validated. And I think that's something that perhaps the research community needs to take away and think about and work out how we best implement that without boring graduate students to death.
I think the other thing that struck us was the great strides that the suppliers are going to improve validation, and I'll try and look around and quickly see the great talks. And I think both in the amount of effort that's going on, but also the kind of public commitment to transparency has to be a good thing. So I think that was a definite sort of one of our common themes, if you like. And then this question of a minimum standard, which is a long discussion, and we're not going to try and summarise that here, but pass it over to the GBSI to sort out in Asilima. And just to say, hopefully, I think that a lot of the discussion here will help shape those standards. So, and then finally, we've a few thanks. So
if, first of all, I think the absolute star of the show has been Laura, who has just so well organised us all. Queries to last-minute changes in programmes to just dealing with the general chaos that is Andy and I. So well done. Very much.
No, it's. Again, I'd just like to echo that because Laura took over halfway through the organisation, so missed loads of the build-up, started a new job and was told, just continue organising this small meeting, which was only about 100 people, and she did it faultlessly, and without that, my life would have been a nightmare, and this meeting would have been nowhere near as successful, so thank you. Also want to thank Matt and Reese from SciTab. So Matt's been involved really early on, and Matt built the website and kept that up to date, which is a sort of ongoing task. And they've both been great at helping behind the scenes and doing everything from like stuffing delegate bags to making sure it runs smoothly at the do. And that's both Matt and Reese, who's only been with us for about 10 days. So he's had a sort of baptism of fire, but has been a brilliant at helping out. So thank you both. But you've got to wait 4 minutes till the afternoon. So, and then just really quickly, just to thank all the sponsors without whom the meeting couldn't run. It was brilliant to see them engaging in this way. and most importantly, to thank all of the speakers, the session chairs, and everybody who's asked questions and made it such an interactive meeting.